Welcome to this learning episode on real-time RT-PCR based coronavirus test. COVID-19 caused by severe acute respiratory coronavirus 2, also called SARS-CoV-2. Most common symptoms of an infected person are cough, fever, difficulty of breathing or shortness of breath. Most suited testing specimens are 1. Nasopharyngeal swab, or 2. Oropharyngeal swab. Collection of nasopharyngeal or oropharyngeal specimen by swab and placed into the viral transport medium. Then, taken to laboratory for viral RNA extraction. The specimen is mixed with lysis buffer containing highly denaturing phenol and guanidine isothiocyanate. Lysis buffer also contains renase inhibitor that prevents the breakdown of viral RNA. After mixing with lysis buffer, the sample is mixed with gentle mixing or false vortexing and incubated at room temperature. And virus is lysed. Lysed samples are loaded into spin column and centrifugation is performed. This centrifugation step is known as solid phase extraction method. Under optimum pH and salt condition, RNA molecules bind to silicon gel membrane. Protein or other contaminants are not retained within the membrane, and centrifugation is done. After centrifugation, the spin column is transferred into a new collection tube and the filtrate is discarded. Then wash buffer is added. Then centrifugation is performed, which removes any remaining impurities, leaving only the RNA bound to the membrane. Then the spin column is placed in a new microcentrifuge tube and elution buffer is added. And centrifugation is performed again, by which viral RNA is eluted from the spin column. Then purified RNA, free from proteins, contaminants and another inhibitor are obtained. Preparation of master mix, PCR reagents and placement into PCR machine, for the preparation of reaction mixture. Master mix containing, reverse transcriptase, dNTPS, TaqMan probe, DNA polymerase, forward primer, reverse primer, and finally, viral RNA template is added. After gentle mixing, reaction mixture is taken into PCR tube. Then, placed into PCR machine. During the first cycle of polymerase chain reaction, the first step is reverse transcription of viral RNA to complementary DNA. Second step is initial denaturation, causing separation of RNA-cDNA hybrid. In the annealing step, annealing of primer to the single-stranded DNA template occurs. In extension step, DNA polymerase synthesizes new DNA strand, complementary to the template. Finally, double-stranded DNA target is obtained. In the second cycle of polymerase chain reaction, denaturation yielding two single-stranded DNA. Next, annealing of forward and reverse primer, and TaqMan probe to its complementary site. TaqMan probe containing 5' fluorescence reporter, that emit fluorescence when excited, and 3' quencher, which prevent fluorescence emission. Then, extension of DNA polymerase, cleaves the probe, separating the dye from the quencher, and emit fluorescent light. In each cycle of PCR, more dye molecules are released. Increase in fluorescence intensity, which is proportional to the amount of amplicon synthesis. Light from the tungsten halogen lamp passes through extraction and reflected by mirror and fall on the dye molecule. Light from the excited fluorophore passes through the emission filter. That is detected by charge coupled device camera or CCD camera, which converts the light that it captured as an individual data. Fluorescent light capturing as individual data. This method is known as real-time PCR, which allows the monitoring of the progress of PCR reaction.